So the phage therapy was eventually abandoned pretty much in the early 40s with the introduction of the first chemical antibiotics. And they were much more reliable. They had a broad spectrum. The principles on which they acted were better understood. They had much more predictable pharmacokinetics. And also we didn't have to have a very good understanding of the phage pathogen interaction in order for it to work, because it had such a broad spectrum of activity. So with phage, you have to have a pretty good idea of the actual bacteria you're treating if you're going to use a particular phage. So, and also particularly, this is something which is a little bit, is kind of a theory, which is that politics had a big effect also, because one of the other big early champion of phage therapy was Stalin. And so there was, I believe there was kind of a feeling in the West that, Stalin really likes it, probably not very good. So, and so that the West really, they tried to get away from this kind of Soviet style, scary Soviet medicine to go with a more defined chemical. Although the Soviets also continued to use antibiotics as well. So, this is a Max Delbruck moment. This is an interesting story about how well the biology of phage was understood at the time, and also it's an interesting story. So, I believe in the 30s, Max Delbruck visited a lab where they were producing these therapeutic phages. And so the product which they were selling was supposed to contain 10 phages. And the technician, so he went and talked to them and asked how they produced it. He talked to the technician there, and the technician said, well, to save time, we used to grow them all individually and mix them together, but to save time, we just have one culture, we just mix them all together. And the culture would let all 10 of them grow up at the same time. And when Delbruck sampled the output of that culture, it was dominated by a single phage, and that phage is T7, and that is what it's still now known as T7. So that's where T7 came from. So in the Soviet Union, where Stalin was championing phage therapy, and of course Stalin liked it, you better also like it if you live in the Soviet Union. So there was a lot more work done on it in the Soviet Union. There was an institute founded in Tbilisi, which is now called the Eliava Institute. I believe it was originally called the Institute for Vaccines and Therapeutics in Georgia, which is, of course, Stalin's home country. Tbilisi is his hometown, so that's where it was funded. So they continued to develop it from the 20s, and the center is still there. It still operates, the phage therapy center. We hit hard times after the fall of the Soviet Union, and most of their equipment either broke or was stolen. But they're still there, and the people are still there making product. So the reports from Soviet phage therapy were more favorable. They continued publishing up until now. Of course, there's a problem in the West here that most of it was written in Russian. That makes it less accessible. There are some English language papers, particularly there's a group in Poland that in the 80s that tended to publish mostly in English. And so those papers actually are available and have their clinical trials. And they do report pretty high efficacy, about the equivalent of what you'd expect with antibiotics. Usually non-cure rates in the 80 or 90% range. Now, the problem, of course, it's not proper placebo double-blind, placebo-controlled double-blind experiments that they would run. They would often they would compare a new phage preparation to an old phage preparation, or a phage preparation to antibiotics, or they would treat people who had been treated by antibiotics and had no cure, and then they would go with phages and mix all over, and the phages cured them. But in the paper, it's good anecdotal evidence, but it's often it's not highly rigorous by Western standards. And also, cure was typically evaluated on kind of a clinical level rather than bacteriological level. There's usually no record of any follow-up that was done with patients. So the work is certainly encouraging, I think, for working on phage therapy, although it's not exactly conclusive. So here's a picture of Durrell. So actually, Durrell went to the Eliava Institute in the 30s, but before the Second World War, the Soviet Union wasn't the great enemy of the West. It was the country. So people would go there. So this is a picture of Durrell. I believe this is in the late 30s. This is an unknown technician, typical, right? And this is George Eliava, who the center is now named for. And so Eliava was kind of the driving force behind early Soviet phage therapy, well, that didn't save him from getting killed by Stalin when Stalin had some purges in the late 40s, and he was still, pretty much taking a back shot. So it's pretty unfortunate. Of course, Harel had left before things like that had happened. So fast forward now from this early period to now. So why is there now interest in using phages as antimicrobials? So probably the biggest reason, which is in the media all the time, is the increase in antimicrobial resistance in bacteria. So this is just an example with Staph aureus, methicillin-resistant Staphylococcus aureus. So MRSA, it carries the methane cassette, so it'll be resistant to all lactams and cephalosporins, and they also typically carry some other cassettes that give them resistance to other drugs as well. So they are treatable with a few drugs. Vancomycin is still usable. Linezolid, tigecycline can be used, but these drugs all have side effects. So they're not favored. And so if you look here, so they have the increase in prevalence. This is 3 different surveillance systems. One surveils only ICU, one surveils soft tissue wounds. But you can see a general increase in all. And in some cases, it's up over 50% of the infections are resistant to methicillin, which means most of the frontline antibiotics are not going to be useful in those infections. So, and at the same time, there's been a drop in the number of new antimicrobials that are being developed by the pharmaceutical companies. So this graph here shows the in five-year intervals, the number of new antimicrobials that have been released. So we're down to 4 in the last 5 years, which are linezolid and tigecycline and a couple of others. And the pharmaceutical industry is not apparently very interested in developing any new antimicrobials. According to a recent report, the average cost for drug development now is $800 million. That includes development and taking to clinical trials. And that's the cost of supporting the market. I imagine marketing, we'll probably spend another $800 million in marketing, but you don't get the same kind of cost recovery from antimicrobials as you would get, say, for something like Lipitor, where they're going to take it every day for 20 years. And antimicrobials are almost always used as acute care therapy, so somebody's going to be on them for only a couple of weeks. So the cost recovery is not very good. And so this has caused a real movement out of small molecule antimicrobial development in the pharmaceutical industry. So these are some examples of some modern phage therapy studies that have been done. By modern, I mean, say, since the 80s. So these are you can find all these papers in PubMed, and I can also give you the citations if you like. So different things. So this is a series of papers of their clinical results from Poland, which cover I think a grand total of almost 1,000 patients. So those came out from 83 to 87. Soothill in the UK, who treated both Staphylococcus and Pseudomonas in mice and dogs. There's a famous paper by Smith and Huggins in 82 where they treated E. coli diarrhea in calves. This experiment by Merrill, and this is a long-circulating phage experiment we'll talk about in a bit, but they were able to treat E. coli bacteremia in mice. Cerveni here, they did treatments in mice in Vibrio prolificus, which is a bacteria that infects people who have problems with high iron in their blood, and it's carried by shellfish. This work here with Staph aureus in mice in a wound model, Enterococcus faecium in a mouse wound model, and this is a vancomycin-resistant Enterococcus, which is also another major concern in surgical wounds. And most recently here is some work with E. coli 15787 clearance in sheep.